It's 4:32, so I think we're going to get started with the seminar. So this is the second in our series of the IGERT program on ocean change seminars. Dr. Malin Pinsky is here. We'd really like to thank both DeSalle and NSF for the funding to hold this seminar series in the winter. And it's my great pleasure to introduce Malin Pinsky today, who's going to be giving our seminar on impacts of climate velocity on marine communities. Broadly, his research focuses on the responses of coastal marine communities to climate change and variability, both in temperate North America and on tropical reefs of the Western Pacific. A lot of his work uses long-term ecological data sets or population genomic methods to answer questions at larger spatial and temporal scales than are typically addressed in ecology. He's also very interested in increasingly interested in coupled social ecological systems and ecosystem services. He's currently an assistant professor in the Department of Ecology, Evolution, and Natural Resources at Rutgers University, and an Alfred P. Sloan Fellow in Ocean Sciences. Previously, he was a David H. Smith Conservation Research Fellow at Princeton University, and he received his PhD in biology from Stanford, as well as getting his undergraduate degree in biology and environmental studies from Williams College. So without further ado, I'd like to introduce Malin Pinsky. Thank you.
This afternoon, had a wonderful day hearing about some of the research that's going on here. It seems like a really fantastic IGERT program that you guys have. So, I guess one thing is the microphone on. I think it is. I just need to switch over to my talk. Okay. So one of the questions that I'm really interested in is what climate change is going to mean going forward for the diversity of species around the world. And at a really basic level, species have four ways they can respond to changes in the environment. They can colonize new territory with more favorable environmental conditions. They can use phenotypic or behavioral plasticity and adapt in place. They can evolve, or if all of these things fail, they can go extinct. I think there's some really interesting questions about how, across the diversity of species and diversity of ecosystems, these different coping strategies will play out, and what this means for communities going forward. One of the most commonly observed responses to changing climates has been shifts in species distributions. This graph shows just a couple examples. On the left, 3 species of fish in the North Sea moving towards the poles at different rates. And on the right, a broad diversity of small mammals in the Sierra Nevada. Some of them moving towards higher elevations. On average, they were moving to higher elevations. But one of the conundrums, when people have looked across a wide range of different data sets, is that up to 60% of species are not moving as you might expect from global warming, meaning they're not moving to higher latitudes, higher elevations, or in the ocean towards deeper depths. And at least one of the conclusions from this has been that species' responses to changing climate are fundamentally very idiosyncratic and probably unpredictable at some level. Going to depend very on details of life history and physiology and species interactions and local environmental effects. But I think this creates a really interesting challenge about what aspects of climate change and climate variability actually are predictable, and are there certain scales of biological organization that may be more predictable than others. So I think one of the exciting challenges is to understand both how changing environments and changing climates are affecting local ecological and evolutionary interactions between species and species in the environment, but then how these scale up to broad patterns, population dynamics, dynamic biogeography, and then ultimately what this means for human societies and the kinds of ecosystem services that we often rely on from these ecosystems. Realizing as well that there are really important feedbacks. We're also our behavior affects these ecosystems a lot and also changes local ecological and evolutionary interactions. So the research in my lab tries to work across these scales at local scales materials. We use population genomics to understand the interaction between local adaptation and climate, as well as the demographic processes that are driving metapopulation and meta-community dynamics. And then we use long-term ecological data sets and comparative data sets to understand how these local processes scale up to regional and global patterns, including changes in abundance and changes in biogeography. A lot of the talk today is going to focus on this broader scale work. So when we think about what changing climates mean in the ocean, one of the comparisons that pops out immediately when we think about what's happening on land is that there's been much slower warming in the ocean, shown here in the blue lines, than we've seen on land, which is in the orange and red lines. On the other hand, marine species are also adapted to much narrower ranges of temperature variability. So these two graphs show ranges of seasonal variability on the y-axis. And then the x-axis is latitude on land and in the ocean. So even what this may mean is that even a small change in temperature means a lot for marine species relative to historical or even evolutionary experience for different lineages. In addition, spatial gradients in temperature are much shallower in the ocean. So if you want to see a half-degree change in temperature, on land, on average, you've got to go about 50 kilometers. In the ocean, at least in the surface ocean, you have to go about 150 kilometers. And where that becomes especially important is when you're thinking about climate velocity. So I just want to explain for a second what that is. You think about any spatial gradient in temperature. For example, it's warmer at the equator, cooler at the poles. If you pick a particular temperature, you could draw that isotherm on a map as a line. And if as that gradient warms, that isotherm is going to move in a particular direction and at a particular speed that you could give a heading and a speed in, say, kilometers per decade. And that combination of direction and speed is climate velocity. Because spatial gradients in the ocean are so shallow, climate velocities in the ocean are as fast, if not faster, than we see on land. So we've got climate velocity. This is from a paper published by Mike Burrows and colleagues a few years ago. Climate velocity on the y-axis, again, across a range of latitudes. For the ocean in blue and on land in red. So even though absolute rates of warming in the ocean are slower than we've seen on land. And in the same paper, they also looked at climate velocities globally for the last 50 years. And in this map, areas in white show climate velocities near zero. Areas in dark orange are climate velocities upwards of 200 kilometers per decade. So we've seen very rapid climate velocities in recent history. But there's also a lot of variability from one part of the world to the next. And while we're at the point that we can map this kind of mosaic of climate velocities, we don't have a very good understanding for what this actually means for species and communities. So I think this raises a lot of really interesting questions. Some very basic questions, including, do species actually follow climate velocity? Is this meaningful from a population perspective? Can species keep up with rapid climate velocities? And are there important limits to how fast species distributions can move? Have we reached those limits? And then how does climate and climate velocity interact with other stressors that are affecting many of these populations? Climate is by no means the only impact on population dynamics. So just focusing on this first question, ideally we want a data source that covers not only a wide spatial area, but goes back, covers a large temporal span, and covers a wide taxonomic diversity of species to try to understand whether species are following climate velocity. And at least in the ocean, we potentially have a fantastic data source in these bottom trawl surveys that have been going on in many parts of the world, including North America, for the last few decades, covering large parts of the continental shelf. They work by dragging a large net along the bottom, often a stratified random design, used for managing fisheries, but also useful for a large number of other analyses. And then they count and weigh a wide diversity of fish and invertebrates that they pull up. So one of the things that colleagues and I have done is pulled together and compiled bottom trawl surveys from many different parts of North America, regions shown here in red, from the Bering Sea down to the Gulf of Mexico and up to Newfoundland. And to summarize a lot of data analysis rather succinctly, we can show the center of a species distribution as captured in these surveys as a dot on a map. In this case, American lobster in the northeast US. And then show how the center of that distribution has shifted over the, in this case, this is for the late 1960s. You can show how the distribution shifted over the subsequent 4 decades as an arrow on a map. In this case, lobster shifted to the northeast at about 70 kilometers per decade. Turns out that's one of the faster northward shifts that we've seen in this region. And if you look at a wide diversity of species that are caught in this survey, each of the orange arrows here, you can see that some species are shifting north, some are shifting south, some very quickly, and some actually quite slowly, not moving much at all. The average across the entire assemblage is 20 kilometers per decade to the northeast, which, in a broad sense, seems to match the warming quite rapid warming that's been seen in this region. So we can look at not just one region, actually, but a wide number of regions around North America. Again, each of the colored arrows is showing an individual taxa within a region, and the black bold arrow is showing the average across the assemblage that's caught in the survey in each region. And we see that, yes, there are some places where assemblages have been shifting north, including Bering Sea, Northeast US, Newfoundland, but that's by no means the dominant pattern. There are also a number of regions where assemblages have actually been shifting south over the period of these for which we have surveys. And if we compare those shifts in assemblage positions to changes in temperature, you can plot that on a graph here, where on the y-axis we're looking at whether the assemblage was shifting north or south and how quickly in degrees latitude per year. Each of the dots here is showing the average for all the species within a region. Then on the x-axis, we've got change in bottom temperature in degrees Celsius per year. If you're willing to squint a little bit, maybe you'd say that in regions that have been cooling, down the lower left, we see assemblages shifting south. And in regions that have been warming, we actually see assemblages shifting north on average. But there's also a really interesting outlier here, which is the Gulf of Mexico, where it's been warming actually pretty rapidly over the last few decades. But actually the assemblage has been shifting south over that time period. On the other hand, if you start thinking about the geography in which these species exist from a marine perspective, if they headed north, they'd end up in Louisiana. So that's not a viable option. So given the geography, moving north is not a way to find cooler temperatures, but actually moving south towards deeper waters is. And in this case, the assemblage is not only moving south slightly, but also moving towards deeper waters. And that's actually a pattern we've seen across a number of different assemblages, where a number of different regions, cooling and moving deep, warming and moving deeper. So if you're willing, in this particular graph, if you're willing to drop Gulf of Mexico as an outlier, we can explain 60% of the variation across these different regions based on just change in bottom temperature. Turns out we can also, as I mentioned, explain changes in depth as well. That graph right there. So explaining 80% of the variation across these regions based on how fast surface temperatures are changing. In places where surface temperatures are warming, assemblages tend to be moving deeper at a faster rate. And where they're cooling, they're moving towards shallower regions. So even though it does seem like regional assemblages may be tracking these changes in temperature and that there are important differences among regions, so a lot of this is a climate variability signal, this doesn't tell us necessarily why individual taxa may be moving at different rates or moving in different directions. So to do that analysis, we're starting to look at individual taxon-specific climate velocities. We've done that by looking at the distribution of species across the seascape. And this example is for silver hake in the northeast US. Each of the black dots on this map is proportional to the biomass that was caught in the bottom trawl survey in a particular location. There are small gray X's in there where silver hake weren't caught. From this, we can define a realized thermal envelope and then project that back onto the spatial distribution of temperatures that we're seeing across this seascape. And we can project those temperatures across different time periods and ask whether changes in the location of the thermal envelope for these species are matched by changes in the distribution of the species as well. In this particular case, it looks like the movement of silver hake away from the inshore part of the mid-Atlantic bight was mirrored quite well by a shift of the thermal envelope offshore at the same time, between the 1970s and the 2000s. But rather than doing that just for individual species, we've done that across the 325 different taxa for which we have data. And in this case, the x-axis is showing taxon-specific climate velocity. So the rate in degrees latitude per year that the thermal envelope for a species was shifting either south or north. And then on the y-axis, the rate at which the species distribution itself from the surveys was shifting south or north, again in degrees latitude per year. So each of the gray dots in this figure is a taxon within a region. You see a lot of taxa showing up in the upper right. Climate velocities were pointing north, and taxa were moving north. And a lot of dots showing up in the lower left. So climate velocities were pointing south, species were moving south. Since both axes are measured in the same units, we can show the one-to-one line here. And we see that broadly, these shifts that we're seeing in species distribution are clustered around this one-to-one line. Obviously, there's still a lot of variability. On average, we can explain about 40% of the variation across these different taxa based on this one factor of climate velocity. Which I think is a really interesting result. It does suggest that species do follow climate velocity to a certain extent. I think it also suggests that a lot of the variability that we've seen among taxa may not necessarily be unpredictable, but may at least partly be due to the spatial mosaic of climate velocities that we see. There's a lot of climate variability, a lot of variation from one place to the next, and species seem to be following that variability to a large extent. So I think there are also interesting questions about whether species can keep up with climate velocities. So that figure I just showed you provides one way to answer that question, because we can ask whether thermal envelopes are not only shifting south or north, but whether taxa are keeping up with it. So taxa in the lower right-hand corner are lagging behind, meaning they're not shifting north as fast as you would predict just from the thermal envelope. You can measure that distance as a residual. Similarly, any dots that show up in the upper left are not shifting south as fast as you predict from climate velocity. Again, lagging behind. And the opposite for species that show up in the other quadrants, meaning they're speeding ahead. On the other hand, if you put all those residuals into a histogram and ask whether on average are marine species lagging behind, the answer is no, they're not. There are some that are lagging behind, others that are speeding ahead. The average across all these species is not significantly different from zero, which I think is actually a really interesting result in some ways because it contrasts with what we've seen on land, where across small mammals, birds, trees, plants, butterflies, an increasing number of examples where it does look like terrestrial species are lagging behind, meaning that they can't shift their distributions fast enough to keep up with climate velocities. There are many more barriers to dispersal on land, fewer barriers in the ocean, and potentially greater ability to disperse fast enough to keep up with climate velocities in the ocean, though it's something to be tested. We can also ask this in a more nuanced way by asking, are there certain types of species that may be more likely to lag behind or speed ahead of climate velocities? Each of the species in this data set, obviously we know a fair bit about these species. Nearly 200 species of fish, about 125 species of invertebrates. Just to show you results for analysis we've done looking at the fishes, we can ask whether the rate at which these species are shifting, degrees latitude per year, is not only explained by climate velocity and details of the survey, but also by characteristics of the species, including whether they're demersal or pelagic, so living near the bottom or up in the water column, so proxy for adult mobility, whether they're commercially fished or not, and is their biomass increasing or decreasing, how large they get, again potentially a proxy for mobility, how fast individuals grow, which is decently well correlated to population growth rates, how big latitudinally these ranges are, potentially a proxy for phenotypic plasticity, and then where in the food web they're feeding. So we can put all of this into a multiple regression, simplify it down with Eke-Ike's information criterion, and actually what we're left with is a very simple model. Saying that yes, it's climate velocity and some characteristics of the survey that matter, but also growth rate. Which is interesting because it suggests that slow-growing species are more likely to lag behind than fast-growing species, which matches up well with what we'd expect from invasion biology, which says that it's not only dispersal distance that matters, but also the ability to colonize new territory and grow quickly, and then continue colonizing new territory after that. On the other hand, with this model including a species characteristic, we can explain 42% of the variation across this data set. If we were to drop the one life history characteristic in here, we'd explain 41%. So this is not a very large effect. So interesting questions about whether maybe we're not capturing the right metrics of the right species characteristics, or maybe there are other ways to look at this data set. On average, though, it does seem like these lags in marine species so far are relatively minor. So I think raises some interesting questions about what demographic mechanisms may be driving that. I mentioned dispersal as one potential. Most marine species have a larval phase where they're floating with ocean currents for days or months before they settle down to adult habitats. So they have the potential to travel very long distances, though we actually have very poor estimates of how far they actually travel. Partly because there are millions of larvae, nearly all of them die, they're microscopic, so hard to track using traditional methods. So we actually don't have very good estimates for what typical dispersal distances are for most marine species. One approach, though, is to use DNA as a natural tag to understand where larvae have come from. And I'm going to show you one approach that we've taken, studying this species right here, yellow-tailed clownfish. It's a common coral reef species across the Indo-Pacific. And what we can do is we can look at how genetic distance here on the y-axis between populations is related to geographic distance between populations here on the x-axis. And if we sample across a transect that's a couple of 100 kilometers long, we see this pattern where sites that are nearby to each other have a low genetic distance between them. Sites that are farther apart have a higher genetic distance. And overall, there's this classic isolation, what's called an isolation by distance pattern. We did this study on two different islands, so the red and the blue dots and lines I'm showing you here. And the slope of this line actually tells us something about how far larvae are dispersing, how far away they're ending up from their parents. We have to combine that with estimates of how strong genetic drift is in these populations and what the densities of these populations are along these islands. But with that information, we can estimate that, at least in this species, in this geography, larvae are ending up, typical dispersal distances are about 11 kilometers. And with some assumptions about how dispersal falls off with distance, about 95% of larvae are landing within 20 kilometers of where they were spawned. So for a species, actually, in this case, that only spends about 10 days in the plankton, doesn't actually spend a very long time dispersing compared to most marine species. This seems to be a fairly long distance. And I think part of what we need to do going forward is dig into these demographic mechanisms to understand what the limits will be going forward as we expect climate velocities to continue increasing into the future. So I think that we can also look, and we can dig into mechanisms, we can also ask about consequences. So marine species may not be lagging behind climate velocities. So what does this mean for people and fisheries that may be relying on many of these species that seem to be moving very rapidly? So I'm just going to show you one example from a species in the northeast U.S. that's moved north very rapidly. This is red hake. It's a species found near the bottom. Used to be caught in very large numbers, now actually not caught now the fishery is much smaller. But I'm showing you latitude on the y-axis, year on the x-axis from the late 1960s through the 2000s. And the black line is showing how the center of the red hake distribution has moved north from being centered off New Jersey in the late 60s to now centered off Massachusetts. It's one of the species that's shifted north most rapidly in the northeast U.S. We can plot the same sort of thing, though, for the red hake fishery, which also has moved north over the same period of time. So this is the red line. This is the center of distribution for red hake landings by the fishery. But I think that sort of the general northward trend actually obscures sort of a more interesting story here. which is that the red hake fishery seemed to shift north at about the same rate as red hake populations did until 1990. And then after 1990, red hake continued moving north into Massachusetts, but the fishery didn't. And it turns out actually that this fishery uses, red hake are not all that big, they get up to sort of 50 or 60 centimeters. And the nets that are used to catch them have a relatively small mesh size and are prohibited from many parts of northern New England out of concerns that they'll catch other species that are not doing that well. So it's actually relatively hard to go fishing for red hake in Massachusetts and further north. In addition, there aren't many markets for red hake up in Massachusetts. Turns out the fish doesn't travel very well. You can sell it to New York City and Fulton Fish Market, but up in northern New England, no one really wants to buy it. So for a range of regulatory, economic, and social reasons, the fishery has been lagging behind the fish as they've moved north. On average, that, if you want to summarize that, it's been about 75% slower. We've looked across a few different species that have shifted north quite rapidly, and we've seen this in each of these species. Not only red hake, but also summer flounder, American lobster, and yellowtail flounder. And one of the things that we think may be going on, though we're just starting to dig into this in collaboration with some colleagues who are anthropologists and economists, is that this interaction between the social system and how fishers are reacting to these shifts, and then their impact back on the population may help explain why some of these species are shifting north so quickly. So if we think about, just in a very cartoon sense, there's a species distribution. We've got that's being pushed by climate velocity to the right-hand side of the graph. If the fishery is focusing on the trailing edge of this population, reducing abundance there, but releasing the leading edge of the population, where the population can actually grow more quickly. That may actually help colonization at the leading edge, but also may help explain why the center of biomass, which is the metric we've often been using, may be shifting faster than climate velocity. Because you've got these differential effects of fishing at different parts of the range. And some of the work that we're starting to do going forward is trying to understand how actually the rules under which fishing takes place and how institutions structure the incentives for fishermen to adapt to these kinds of changes. So at least what we know right now is that it looks like on average marine species aren't lagging behind all that much, but fisheries do. And now trying to understand the more complex social ecological dynamics, what this means going forward for the sustainability of fish and fisheries in these systems. So for the last bit of the talk, I want to think about how climate interacts with other stressors in the marine environment. And one contrast we can make between what we've seen in the ocean and our experience on land is illustrated in this graphical timeline from a review some colleagues and I published just a couple weeks ago, where you see on land that widespread hunting of terrestrial animals started tens of thousands of years ago, but has largely transitioned to habitat transformation on land. Agriculture in cities really transforming habitats for a wide range of species. Whereas in the ocean, human impacts have lagged behind substantially. So widespread exploitation is maybe a century or so old, and we're only just beginning to see habitat transformation really take off. And what that has meant is that at least the extinctions we know about in the ocean have occurred much more recently in time than the ones we know about on land. Human-caused extinctions on land started more than 30,000 years ago and have really accelerated in recent years. We know of 500 extinctions in the last 500 years on land, whereas in the ocean, we at least know of only 15 extinctions over the last 500 years that have been caused by humans. On the other hand, we also seem to be seeing this transition from hunting being the dominant human impact on a wide range of marine species to habitat loss potentially becoming a much more important factor going forward. These graphs show sort of a summary of some of the changes we've seen, from increasing loss of coral cover in the upper left, similar patterns for mangroves, and then also metrics of human transformation of habitats. very rapidly growing numbers of wind turbines out in the ocean, over here. Seabed mining claims also rapidly increasing, and seabed mining actually really starting to become a practical reality in places. Container ship traffic also increasing dramatically with impacts on the acoustic environment, as well as ship strikes, pollution, and transfer of invasive species around. And then also number of dead zones around the world also starting to increase dramatically. And I think one of the lessons we've learned on land is that, and in the ocean, is that species can rebound relatively quickly from hunting in many cases if hunting or fishing pressure is reduced. But habitat transformation tends to be much harder to reverse. On the other hand, what we've seen, still I think fishing right now is the dominant human impact on many marine species. And I want to show you a few examples of how we've been trying to think about the interaction of fishing with different aspects of climate change and variability. Not only climate velocity, which is what I've been talking about so far, but also climate variability and potential ability to adapt to future conditions going forward. So if we think about how climate velocity interacts with fishing, one way to think about this in a cartoon way is that you've got a thermal envelope for a species, shown here in red, and that's going to move across space. And the question is whether the population, the fish, can keep up with that thermal envelope while it's being fished while it's being harvested. So one of the ways to at least build some intuition about how this process works is to put this together into a mathematical model. Put some assumptions in, see what comes out. This is work I've been doing with Emma Fuller and Eleanor Brush, two PhD students at Princeton. Those of you who want the gory details, and it's a spatial population dynamics model and an integra difference model. But a way to summarize the results from that is on this figure here, where we've got a harvest rate increasing on the y-axis from low to high. The absolute numbers don't matter, and climate velocity increasing from 0 to some higher number. And what we see is that in this parameter space, there's this curved boundary that divides two regions, where at low harvest rates or low climate velocities, you expect populations to persist, and at high climate velocities or high harvest rates, you expect populations to go extinct. The exact location of that boundary depends on the kinds of species characteristics you'd expect. How far they disperse, how fast they grow, the shape, to a certain extent, the shape of the dispersal curl as well. But one of the important takeaway messages is that this is a curved boundary, and that tells us something about the interaction between these two stressors. So for a given harvest rate, there's a maximum climate velocity that a given species can keep up with. But at a higher harvest rate, the maximum climate velocity they can withstand is going to be lower. And part of that seems to rely on whether or not we fish the leading edge of a population. So if you fish the leading edge, you're reducing the growth rate at that leading edge, which, as I mentioned before, ties back to invasion biology. If the growth rate at the leading edge is lower, species can invade territory at a slower rate. So this has some interesting parallels, again, to what we've seen on land, where habitat fragmentation is also a cumulative impact that tends to slow species' ability to move across the landscape and keep up with climate velocities. But on the other hand, but for a very different reason. Rather than reducing growth rates, it tends to impede dispersal. So similar outcome, but different processes underlying what's going on. So one thing you might say is, to help species keep up with climate velocities, why don't we put up a network of marine protected areas across this landscape? You can say that you can't fish in certain places, and ask whether, at least in this toy model, whether that makes a difference for the ability of species to keep up with climate velocities. And I'm going to show you the results in a very similar figure to what I showed you before. Again, we've got harvest rates increasing on the y-axis, climate velocities increasing on the x-axis. I'm showing you equilibrium biomass as shades of gray, but it's the same idea. White being higher biomass, black being extinction. So this is without protected areas. This is our base case, what you'd expect. And then with protected areas, we see that the range of parameter space, the range of cumulative impacts from these two stressors becomes broader, under which this population can persist. So this boundary between persistence and extinction is moving up and moving out. So it seems like protected areas could help quite a bit. But on the other hand, that's under one particular set of assumptions about how humans react to protected areas. That's not the only assumption you could make. You might say that rather than being eliminated, fishing effort, rather than being eliminated from protected areas, it's actually just displaced to the areas outside the protected areas. So you end up with actually a stronger mosaic than you had before, where you have intense fishing outside and no fishing inside. In essence, fragmenting the landscape to a certain extent, but through harvest rates. And here we actually see the opposite effect. So actually in this case, it's harder for species to keep up with climate velocities if they're moving through a landscape of very intense fishing pressure and then lower fishing pressures. The cumulative impacts, because they're nonlinear, lead to this kind of pattern. What that means, though, is that we really have to understand it. Again, the human side of this equation. Just understanding the ecology won't be enough. But understanding how people and fishers react to these kinds of regulations and spatial management measures becomes an important part of the story. So the other kind of cumulative impact between climate and fishing that I want to talk about is fishing and climate variability. Not necessarily the long-term change, but just the magnitude of variability through time. So this started inspired by relatively, there have been plenty of stories about fisheries that have collapsed to low abundance. We know of many examples around the world. But we also know of many species, including many forage species, that fluctuate a lot in abundance through time. That seems to be tied to variability in the environment, not to necessarily changes in fishing pressure. Sardine and anchovy being a classic case. So we were really interested in whether some of these collapses of fisheries to of fish populations to low abundance may be explained by climate variability rather than by high fishing pressure. So there's thanks to many people in this room, there's something called the RAM Legacy Database. Compiles together stock assessments that record fish population dynamics for many different populations and invertebrate populations all over the world, and allow large-scale meta-analyses to be done. So this map here is providing one summary of that data set, where the areas in red show areas where a large proportion of the populations for which we have information have collapsed to low abundance, meaning they've collapsed to less than 10% of the unfished biomass. We've combined that with information on climate variability in each of these regions. So in this figure, areas in dark blue have a high standard deviation of sea surface temperatures. Relatively crude, a very crude metric of climate variability, but starts to give us a sense. It shows that especially ecosystems at temperate latitudes have a higher climate variability, and small enclosed seas, again, have a high climate variability. Towards the poles and towards the tropics, we see much lower magnitudes of variability through time. We've also pulled in information on fishing pressure and species characteristics to try to understand what can predict the likelihood of a population collapsing to low abundance. One of the results that has come out of that work, and this is the modeling framework we've been using for this, is boosted regression trees, which are actually especially nice for looking at interactions between factors. And this surface right here is showing how the probability of collapse here on the vertical axis is affected by maximum fishing pressure, which is the ratio of maximum fishing mortality divided by the fishing mortality rate that's projected to be sustainable for a given population. And then here, metric of climate variability increasing on this lower left axis. What we see is that for species that have not experienced overfishing, climate variability does not appear to matter at all for their probability of collapsing to low abundance. But for species that have experienced, for populations that have experienced overfishing, moving from a low variability environment to a high variability environment can more than double the probability of collapsing to low abundance. So it looks like fishing may be increasing the sensitivity of these species to climate variability. And there may be a range of reasons for this, including loss of biocomplexity and loss of intraspecific diversity within these populations as a result of fishing. Also some interesting theory about how fishing truncates the age structure and makes populations more sensitive to climate variability. That may be the kind of effect that we're seeing across a wide range of species all over the world. So the last bit I want to talk about is about the ability of these populations to adapt to future conditions. At a really fundamental level, genetic diversity provides the raw ingredients for species to adapt to future conditions. And many populations that have been heavily fished have declined to low abundance. It's possible that they've lost genetic diversity as a result of that. But when people have gone out and actually looked for that effect, there have been a wide range of conclusions. In some cases, it looks like genetic diversity has been lost. In other cases, it looks like it doesn't look like any genetic diversity has been lost from these declines in abundance. On the other hand, these studies have tended to have relatively low statistical power to detect the magnitude of effect that we'd expect to appear. So one of the things we decided to do was take a different approach. Instead of looking before and after, let's look across a wide range of species and compare overfished populations that have declined to low abundance to healthy species in the same species or taxon, and ask whether genetic diversity is different between these pairs. So we pulled together information for 140 different species from published literature across 11,000 microsatellite loci. And one of the metrics of genetic diversity we looked at was change in allelic richness, so just the number of different genetic variants at a given locus. We had data across a range of different kinds of marine fish, including tunas and herring, cod and snappers. And I'm showing you the results. I'll show you the results here on this horizontal axis, which is showing the percent difference in allelic richness between species that are overfished and related species that weren't, or populations that were overfished. Where any dots that fall over here had lower diversity in the overfished populations, any dots over here had higher diversity in the overfished populations. And what we see is that for many taxa, individual taxa, it's hard to tell from the existing data whether they're significantly different from no difference, whether there is a significant difference. On the other hand, in 9 of these 12 groups, we've got lower diversity in the overfished populations. If you just did a simple average weighted by the variance for each of these groups, you'd see 9% fewer alleles in populations that are overfished. And we also, when we've looked at other metrics of genetic diversity, we also see, for example, lower heterozygosity, so the probability of pulling out 2 alleles from a population that are different from each other. On the other hand, we also know that there are many influences on genetic diversity in these populations. So we can also try to correct for a number of those, looking at effects of latitude and region, length and catch, which are both proxies for unfished abundance in these species, and then some methodological issues. But despite that, we still come up with significantly lower genetic diversity in overfished populations, right around 10%. And so the microsatellites that we looked at presumably have no influence on evolutionary fitness in these populations. But on the other hand, it implies that genetic drift in these populations is low enough to remove some of the genetic diversity that is either under weak selection or that could be important in the future as conditions continue to change. I think one of the most interesting parts of this is that we're seeing these effects in large, very widespread populations, like not at all what you would expect to experience a genetic bottleneck. Very different than some of the very small isolated populations that are sort of the classic bottleneck examples. I think from sort of these 3 different looks at how climate and harvest interact, it suggests that harvest may constrain species' ability to respond to climate going forward. So I just wanted to leave you with a brief look into some of the work that's just ongoing in my lab, including trying to understand whether there are maybe higher levels of biological organization that might be more predictable than individual species responses to changing climate. So trying to understand to what extent communities are being pulled apart and reshuffled by climate velocities. We're also using oceanographic models and population genomics to try to understand whether larval dispersal may be driving some of these distribution shifts and whether that's a really important way in which these species are keeping up with climate velocities. And then finally using transcriptomics and population genomics to try to understand whether fish are locally adapted to some of these thermal gradients. And whether, rather than sort of one large population with a wide thermal tolerance, we actually have many small populations that are each locally adapted to a much narrower range of thermal conditions. So just wanted to leave you with sort of a thought. I've tried to, throughout this talk, I've tried to compare what we see, what we think we may be going on land to what we see in the ocean. And even though the same ecological and evolutionary processes are at work in both these ecosystems, the physics and geography and taxonomic diversity is very different. And I think that may lead to important differences in the kinds of climate impacts, the patterns of climate impacts we see in these two ecosystems. So just something to think about, maybe spark discussion going forward. With that, I just want to thank, I felt really lucky to have a fantastic group of collaborators over the past few years working on many parts of this, group of funders, and then a fantastic lab that's doing much of the ongoing work that I was just talking about. Patrick, Michelle, Ryan, and Jennifer. So thanks to all of you for listening.
So we have about 10 minutes for questions, so anyone who has questions?
Wow.
I'll let you know.
Yeah.
I was thinking about the variability map that you showed, showing that the temperate regions have more variability in temperatures, and wondering if the assemblages in those regions might be more protected from the effects of climate because they're adapted already to some variability, or they might be less sensitive to how the effects of harvest and climate.
I think it's a really good question. And it'd be interesting to try to pull in the information about thermal tolerances, which you'd expect would be wider in those areas. On the other hand, I don't necessarily think that that's driving the pattern that we see, because we're not necessarily looking at changes in variability through time. So these are just areas that are more variable. So, and that's where we're seeing higher probability of collapse. So I think it's a really interesting question and may actually be important going forward. You know, do we expect variability to change, which in some cases we do. And in sort of going back to the climate velocity work, even in these places where we see a lot of variability, we still see species distributions shifting quite a lot. That's actually, you know, most of the regions we've looked at so far are in these sort of temperate latitudes. That tends to be where we have decent data to look at how species distributions have been changing. So it looks like even for these species that may have relatively wider thermal envelopes, they're still shifting their distributions. And part of it may be that, at least on the East Coast, we have a bunch of species that migrate back and forth. So that actually, as adults, they're moving to take advantage of some of that variability through the seasons, either inshore and offshore, or north and south. So yeah, I think it's a really interesting question. In the back.
It looked to me like you were looking at changes relative to temperature, but in the oceans we have multiple stressors that are occurring simultaneously. Are you able to make a distinction between the various multiple stresses in terms of the responses that you are looking at?
Yeah, that's a fantastic question. That's something I've been wondering about as well. So the stuff I showed you, that's right, it's temperature. The physiological mechanism by which temperature is thought to affect growth rates in fish and other marine ectotherms, it's actually probably related to oxygen. Metabolic demand for oxygen goes up with temperature, but oxygen content in the water goes down. So it may actually be an oxygen limitation that really is the mechanism by which temperature affects physiology. But there are also interesting questions about changing oxygen concentrations, changing primary productivity, so productivity in the food web, or just habitat availability as well. As these species shift across the landscape, some of these species depend on hard bottom habitats that may or may not be available in the places that they're shifting into. So this is some of the stuff we're just starting to do. I don't have any results to show you, but I do think, when we look at to what extent are species following climate velocities, that graph I showed you, there's still a lot of variability, and I think multiple stressors end up being a really important part of that question. So the other ecological impacts, including probably species interactions. So the interaction between fisheries and climate velocity is something we've been looking at, but that's by no means the whole story. I think that's a really important part of it. Yeah.
For the climate velocity work, I was wondering if you could look at, or if you could say a bit more about shifts in abundance in addition just to the range shifts. So do you see abundance shifting throughout the range, or do you see potential demographic effects at the leading or trailing edge?
Yeah, so that's a great question. That's sort of a detail I maybe glossed over a little bit. So the metric of species distribution we're using is the center of biomass. So that includes both changes in abundance and changes in distribution. And part of the reason we didn't look at, for example, range edges is that, at least for the species in here that have changed a lot in abundance, we often see ranges expanding and contracting with changes in abundance. So you may see, even if the center of distribution is moving north, you might see a southern range edge moving south because the abundance is going up. So far we've just been looking at the center of abundance, which could be caused either by shifting range edges or just biomass redistributing within that range. So a lot of what we're seeing, I think, are changes in abundance, and sort of that individuals becoming more abundant towards the poles. Great question. Yeah. This side of the room has questions.
So one of the major questions that people ask in global change conversations is, oh, well, can my pet species evolve fast enough to keep up with the trend of change, right? And so you were getting at some of that evolvability, looking at just the stock of genetic variability in some of these species, in particular the microsets in the commercially fished species, right? So I wondered if, given that you have this data set of a couple of hundred species, and I forget what it was several hundred species, right? And 11,000 microsets. Have you thought about just mapping the stock of existing genetic diversity for these species onto your climate map and asking how you basically get at a genetic vulnerability versus genetic adaptability or an evolvability index given the threats that the species face from climate?
Yeah, one of the things we're trying to get a handle on right now is, so we started out with sort of which species were overfished and which close relatives could we get genetic data on. We've expanded that out to as many marine fish as we can get genetic data on. And just trying to understand sort of basic questions about global gradients in genetic diversity. Do metrics of intra-species diversity follow interspecies diversity across the globe? And there's some interesting, in the ocean, there's some interesting patterns where in coastal species you see highest species richness showing up in the coral triangle and tropical latitudes. But in oceanic species, you actually see peaks at intermediate latitudes in both hemispheres. So we're curious whether that's mirrored in genetic diversity as well, whether there may be similar patterns structuring both kinds of diversity at different levels of biological organization. But yeah, it's a really interesting thought to try to think about how that plays out.
Interesting. Jen Sunday's work on ocean acidification of these couple of different species, and just making the point that if you have a bunch of genetic diversity, well, then selection has something to work with. But if you have no standing genetic diversity, there can be no selection.
Yeah, I think in a really broad sense, marine populations tend to be really large. There tends to be a lot of gene flow up and down the coast and across different environments. So I think in general that will tend to give marine species more genetic material to work with going forward in terms of evolvability, but I think there are some really interesting questions that we're just trying to get the tools to look at that kind of question. Definitely, some of the next generation sequencing is starting to make those questions possible.
Is there any role for changes in hydrologic forcing, variability, coastal currents in explaining any of these vectors that you're attributing to temperature?
Yeah, so, you know, some of the, we just started a project trying to understand whether ocean currents may explain some of the variation, you know, where it becomes it's relatively easier to move with ocean currents and harder to move against them. I don't have an answer yet. I mean, part of that will depend on even ocean currents that are necessarily moving in one direction. There's a lot of variation through time, and marine species seem to be very good at taking advantage of eddies that move the opposite direction, or seasonal reversals in currents or undercurrents to even move against sort of mean advective flow. So wait and see sort of how important that is. And in terms of hydrologic forcing, it just depends on the region. You know, a place like the Gulf of Maine sometimes gets called a big estuary because it is so influenced by runoff from rivers all around its edge, and that really can really drive dynamics and also productivity. So the base of the food web.
That's where I was going with it. Is you're getting atmospheric heating, you're going to be changing the distribution of thermal gradients and the deposition of moisture, and therefore the transfer from land to the ocean. So it seems to me that temperature is one of the pieces of the puzzle. I wonder whether you could break out the different species, ones that had, you know, long distance dispersal versus the opposite of long distance dispersal, and see whether they were related to variation in the trajectory of the water.
Yeah, exactly. Yeah, interesting question. We're just starting to try to pull some of those data together. You know, the other thing, you know, as you point out, it's not just sort of the temperatures that determine a given species, but also sort of the productivity of the food web and whether food is even available. And that's a case where folks building these Earth system models that run off global climate models and also regional oceanographic models are starting to get a sense for how the physics is coupled up to productivity at the base of the food chain. Estimates of primary production or even zooplankton production a little bit higher up.
With your, about 2/3 of the way through, you showed that graph looking at the climate variability and the fishing intensity on the probability of collapse.
Yeah.
I guess I'm wondering how much of that response is compounded by the life history characteristics of the fishes that map off across that surface.
Yeah, so there are, I didn't show you the rest of the analysis, so this all goes into a boosted regression tree analysis that includes life history characteristics like growth rate, trophic level, number of eggs, which ends up not being too important, but a number of other traits. Growth rate also ends up being very important. So, though maybe not in the way you'd expect. Sort of traditional conservation biology expectations would be populations with slow growth rates are more vulnerable to something like fishing. We actually see the opposite, that it's actually species with high growth rates are more likely to collapse to low abundance. But it's really that interaction. Actually, again, it ends up being a similar graph to what I showed you for climate variability, where species that are not heavily overfished, growth rate doesn't matter all that much. But species that have experienced overfishing, growth rate matters a lot, with high growth rates being correlated with higher probabilities of collapse. And there's some interesting modeling that folks have done, trying to look at lags in fisheries management and environmental variability. And in cases where the environment is varying through time, and you have a management system that cannot react immediately. You know, it takes a little while to realize the environment has changed, productivity has gone down, and therefore management should be more restrictive and reduce harvest rates. It's actually the populations that have a short generation time and grow quickly that end up being most sensitive to that sort of mistakes in that coupled social ecological system.
But I guess the other thing to consider is that, you know, these species are collapsing based on your definition all the time, aside from fishing pressure.
So yeah, so if that was happening, or if that was a really strong pattern, I mean that's what initiated this research. Is, you know, is this happening just because of climate variability? No impact on fishing. So if that was a really strong pattern in this data set, I would have expected that climate variability would have mattered a lot, even for populations that were not heavily fished. And that's not what we saw coming out of the data set. But
Okay.
good question. Yeah.
So we're out of time, unfortunately, but there's a reception in the lobby, and you can ask Malin questions there with beer and food and stuff. So thank you again.